This insta-lecture session would be on the concept of granuloma and the granulomatous inflammation. I think while reading medicine textbooks, you have many places read, like in Crohn disease, that is characterized by granuloma. For example, Crohn disease is characterized by non-caseating granuloma. In tuberculosis, you see, you often read there is a granulomatous reaction is seen. Sarcoidosis in the lung pathology, you have read that is characterized by non-caseating granuloma in multiple parts of the body. So today we try to understand what is meant by granuloma, why I am so much bothered about this granuloma and granulomatous inflammation. Now, the first concept that is need to be understood is that granulomatous inflammation is a distinctive subset of chronic inflammation. You know that inflammation is a host protective response which is mostly mediated by the vascularized tissue to serve two purposes. One is to eliminate or neutralize the offending agent, the insulting agent, and second to pave the way for healing, tissue healing. Now, there are two forms of inflammation classically seen. One is rapid instantaneous form, we call it acute inflammation. Another is sustained protracted form, which is called chronic inflammation. Now, this granulomatous inflammation is basically a unique distinctive subtype of chronic inflammation. This is the first concept you need to understand. So, in other words, all granulomatous inflammation are chronic inflammation, but all chronic inflammation are not granulomatous inflammation. This is the concept that you need to understand. Now, granulomatous inflammation is called granulomatous inflammation because when, as a pathologist, we look at the biopsy, we see a distinctive microscopic structure which can only be seen in microscope, that is called granuloma. What is granuloma? Granuloma is a unique pattern of tissue reaction which is characterized by formation of some collection or aggregate of some activated macrophages. So some activated macrophages at some place they assemble together, and that those activated macrophages are typically cordoned off by at the periphery by a collar or rim of lymphocytes and plasma cell. So if you get at a tissue someplace there is a activated macrophages which I'm coming to mention, actually they will be called epithelioid cell or giant cell. Those activated macrophages are encircled by lymphocyte and plasma cell. This structure, particularly this structure, is called granuloma. And if this granuloma is seen, this kind of inflammatory tissue injury pattern, which is inflammation pattern, we call it granulomatous inflammation. This is the basic idea. Now, granuloma means it's a microscopic structure which is shows aggregates of lots of activated macrophages. Now, activated macrophages in the granuloma, they take a unique appearance. This is known as epithelioid cell. This is called epithelioid cell because they are not truly epithelial cell, because they are actually activated macrophages. They are not epithelial cell, but they start to look like epithelioid. Anything ending with OID, it means resembling that, mimicking that, like previously discussed about the leukemoid reaction, which is not truly leukemia but looks like leukemia. Similarly, epithelioid cell is not truly epithelial cell, but they resemble the epithelial cell. Usually they have abundant cytoplasm, eosinophilic cytoplasm, and indistinct cell boundary. They look like epithelial cell. This is the typical description of the epithelioid cell. Now, some of the activated macrophages could fuse together and they form a multinucleated cell, we call it giant cell. So what we see in a granuloma, we see collection of the activated macrophages which are taking the form of the epithelioid cell, some giant cell, and they are at the periphery, they are encircled by lymphocytes and plasma cell. And when I think about the granuloma, I usually think it as a prison. Like, why do we keep prisons in the I think in your city there would definitely some prison is there where the criminals are kept. What is the idea of creating the prison or the prison cells? Basically to isolate the criminals from the normal civilians. That is the basic idea, is not it? Because if we keep the criminals along with the normal civilians, they would create havoc, they would create a lot of problem to the normal civilians actually. So to stop that, we isolate them, we keep them in a separate place, we create a prison. Similarly, when the body cannot eradicate, cannot destroy some offending organism which remain persistent in the body despite its best attempt, then the body creates small microscopic structure which are known as granuloma. They are just like a prison or cell, and this prison, those offending organism, the inserting organism, they try to separate it from the rest of the normal body tissue, so to minimize the damage to the rest of the normal body caused by those offending organism or the associated inflammation. This is the basic purpose of the this is the basic reason why the granuloma and granulomatous reaction is seen. Now, what is the significance of granuloma, getting granuloma on the biopsy? It is significant because if we find a granuloma in a biopsy, granulomatous reaction in a biopsy, then our list of differential diagnosis narrows down to a few, because not many conditions are associated with granuloma or granulomatous inflammation. Like thousands of things can cause inflammatory reaction in a tissue, chronic inflammation. But if we get granuloma, then our list of differential diagnosis is selected few. Like it could be tuberculosis, it could be syphilis, it could be cat scratch disease, it could be Crohn disease, it could be sarcoidosis, it could be some fungal infection like histoplasmosis, it could be berylliosis. Only these few things are there, handful of things are there, which usually causes granulomatous reaction. And according to the organ where they are inciting this granulomatous reaction, we can select. Like if we get a ileocecal biopsy, ileal biopsy, in an intestinal biopsy, if we get granuloma and we are getting a typical history of inflammatory bowel disease like Crohn disease, we definitely think about the Crohn disease as the etiology. So if we get a history of which is looking like sarcoidosis, and we start to see the presence of non-caseating granuloma in the lung biopsy or other parts of the skin biopsy or other part of the body, definitely think about sarcoidosis. So in this way, it helps to make a diagnosis. It helps us to diagnose the things easily, because as a diagnostician or as a pathologist, our job is like a private investigator, a detective. When we see a biopsy, we basically think about few suspected criminals, and then we narrow down to the one which is actually causing it. Similarly, if we see a granulomatous inflammation, then we look at the other perspective, the history, which organ we are seeing this granuloma, and then we can narrow down the list of differential diagnosis to a few, and then finally we can diagnose the organism, the cause also. Like if you are suspecting tuberculosis, you can do some microbiological tests like A/B testing or PCR to confirm that it's truly the granuloma is caused by Mycobacterium tuberculosis. Even in tissue biopsy, you can also detect the AFB. So in this way, actually, they help us to pinpoint the diagnosis. That is the basic purpose of this, finding this granuloma and granulomatous reaction. And as I mentioned, that granulomatous reaction, granulomatous disease or disease associated with granulomatous inflammation, there are only notable few are tuberculosis, syphilis, leprosy. In syphilis, the granulomas are usually called gamma. In tuberculosis, those granulomas are actually called tubercle. So they have specific name in certain diseases. And fungal diseases like Histoplasma capsulatum, dimorphic fungi, we can also get characteristic this granuloma formation. In certain vasculitis, we can see granuloma formation like giant cell arteritis, Churg-Strauss syndrome, which is now known as eosinophilic granulomatous with polyangiitis. You can get the granuloma formation. If we get granuloma formation, actually we see in tissue biopsy, we can pinpoint the diagnosis. Now coming to the granuloma is basically divided into two types. One is known as foreign body granuloma, another is immune granuloma. Foreign body granuloma actually is caused by, incited by some foreign bodies, like talc, could be suture, it could be some food material. These things actually can incite a foreign body reaction. This is called foreign body granuloma. And some immune granulomas are typically caused by tuberculosis or sarcoidosis and these diseases, where some immunogenic reaction creates a formation of the granuloma. And so the basic thing we understood, that granulomatous inflammation is a distinctive subset of chronic inflammation, and the usefulness is that it helps us to narrow down the list of differential diagnosis in a biopsy to a selected few. Thank you very much.